In this video, we're going to talk about transport within the human body, specifically into and out of body cells. Certainly, we do have a circulatory system which facilitates long distance transport within the body, but we are focusing here on very small distances and specifically into or out of body cells. And as we're looking at this, there's a few things we have to keep in mind. One, are entities, whether we're talking about ions or molecules, moving down their gradients or against their gradients. If entities are moving down their gradients, it is going to be what we refer to as passive transport. If entities are moving against their gradients, it is what we know as active transport, and that is going to require energy. For this reason, we need to understand the different gradients within the body, that is to say, the concentration of sodium outside the cell versus the concentration of sodium inside the cell. Accordingly, the same thing with potassium. High concentration of potassium inside of the cell versus outside. And lastly, we need to understand the permeability of ions or molecules across the phospholipid bilayer of cells, the plasma membrane. Certain things can cross that plasma membrane without a channel. Others require a channel. Keep in mind that things that require channels to cross the plasma membrane are polar molecules and ions. Okay, so let's just review some aspects of the intracellular fluid versus the extracellular fluid, specifically the concentration of certain ions or molecules. So we've covered this, but I just want to add some numbers to this so it makes a bit more sense. We are comparing the inside of the cell versus the outside of the cell, specifically the concentrations. And the units of measurement we are looking at are milliequivalents per liter. Keep in mind, this is a quantity with respect to volume, so that connotates a concentration. So we look at this sodium, there's 142 milliequivalents per liter in the extracellular fluid, whereas in the intracellular fluid, there are 10 milliequivalents per liter. This is telling us there's a much higher concentration of sodium outside the cell than inside the cell. And potassium, as we would expect based on what we've talked about previously, there's a higher concentration of potassium in the cell at 140 milliequivalents per liter, versus in the extracellular fluid, potassium maintains only 4 milliequivalents per liter. Calcium has a huge gradient showing 2.4 milliequivalents per liter in the extracellular fluid and only 0.0001 milliequivalents per liter in the intracellular fluid. And then, of course, chloride, 103 versus 4 milliequivalents per liter in the ECF and ICF respectively. Certainly we could have the phosphate ions here. There's going to be a higher concentration of phosphate ions within the intracellular fluid, as well as protein anions, negatively charged proteins. Okay, so let's just have an overview of what passive transport is. Passive transport is the movement of ions or molecules across the plasma membrane. Keep in mind, it does not matter what direction they're going, into the cell versus out of the cell. It's still passive transport as long as no energy is needed. And no energy is ever needed when things are moving down their gradients. Diffusion does not require energy. So I said that in probably 3 different ways. Passive transport is diffusion. It would probably be better for me to say diffusion is a form of passive transport, moving from an area of high concentration to an area of low concentration. So simple diffusion and facilitated diffusion are both passive transport. The difference is simple diffusion is the movement of entities directly across the plasma membrane without the need of assistance, without the need for a protein channel to pass through, versus facilitated diffusion requires a protein channel or a carrier. So what kind of things do you think can move across the plasma membrane without a protein channel? And what kind of things do you think need a protein channel? If you said simple diffusion is for really small molecules like gases, you'd be correct. And if you said lipid-based molecules, which are lipophilic and can easily cross the plasma membrane, you'd be correct. Whereas facilitated diffusion is the opposite. These are hydrophilic molecules or lipophobic molecules. Once again, that's saying the same thing. Charged atoms, known as ions, polar molecules, always require a channel. They require some sort of facilitation to get across the plasma membrane, hence the term facilitated diffusion. Okay, so let's just look at a couple examples of facilitated diffusion. Here we have a cell, plasma membrane. Like I said previously, gases can easily move across the plasma membrane. Oxygen is always going to move into the cell down its chemical gradients, really from an area of high partial pressure of oxygen into an area of low partial pressure. We're dealing with gases here, so that's why I'm referring to partial pressure. We'll focus more on that when we get to the circulatory system, but oxygen's always diffusing into the cell. Carbon dioxide is always diffusing out. Those molecules are so small, they do not need a channel. Lipid-based molecules such as estrogen can easily move across the plasma membrane without the need for a channel. That is simple diffusion. Nitrogen can move across, which is a gas, and alcohols can easily cross the plasma membrane via simple diffusion. Okay, so let's just take a look at a channel protein. Keep in mind we have a plasma membrane here. We see the phospholipid bilayer in dark. And in yellow here, this is representing the lipid tails, which is the nonpolar region of the cell membrane. And that's what dictates what can cross and what can't. Polar molecules and ions are going to require a channel. And what I have here is a channel. Channels are composed of proteins. Channels will span the entirety of the plasma membrane. That is to say, directly across the plasma membrane. And they are going to be structured in such a way that either their physical structure or their chemical structure is going to dictate what kind of things can pass through that channel. So here we have what's known as an aquaporin, which is a water channel. There are about 13 different aquaporins in human body cells, and I'm not suggesting 13 in every cell, but throughout our body there's probably 13 different types of water channels known as aquaporins, which are required for water to move into cells and out of cells. Keep in mind, water is a polar molecule. So if we look at this, my question to you is, what direction is water going to go? Because this water channel will let water into the cell, and it will also let water out of the cell. So what is going to dictate the direction water is going to move? We will, in a subsequent video, talk about gated channels, where these channels are open or closed depending on the biochemical state or conditions. But this type of channel is always open. So what is going to dictate the direction of water, whether water moves into the cell versus out of the cell? I'm hoping you guys were thinking about solute concentrations. Keep in mind, water is always going to move to an area of higher solute concentration. And as you know, that's the same thing as saying water is going to move from an area of high molecular water concentration to an area of lower molecular water concentration. And that's what we refer to as osmosis. So the osmolarity, or the measurement of the solute concentration in the extracellular fluid and the intracellular fluid is going to dictate the direction water is going to move. And ideally, the extracellular fluid and the intracellular fluid are what are known as iso-osmotic. Iso means the same osmolarity, which would lead to very little water movement, because if there's the same osmolarity in the outside of the cell versus the inside, water's not going to move very much or at all. And if it does move, some's going to come in and some water's going to leave at the same time. So let's take another look at facilitated diffusion. Now, facilitated diffusion can be things like sodium. Sodium is going to require a channel to cross that phospholipid bilayer because it is charged. These channel proteins oftentimes are very specific to an entity, whether it's a molecule such as glucose or an ion such as sodium or potassium or calcium, chloride, what have you. Now keep in mind, we're assuming that both sodium and potassium have maintained their gradients. So sodium is going to be inclined, if you will, to move down its gradient into the cell through that channel. But one very special biochemical makeup of these channels are bonds or side chains within that channel that carry a charge. And in this case, there's a bunch of negative charges within the cell, within that channel protein. And because the positive charge of sodium is attracted to negative charges, that's going to allow sodium to pass through that channel. So it's the size of the channel which inhibits other entities from moving through, and the charge, the negative charge, would inhibit chloride from moving through. So we also have glucose that moves through channels and potassium that moves through channels. Keep in mind here, I'm just putting one channel for both glucose and sodium. It doesn't necessarily work that way. Oftentimes they are totally separate channels, but to be clear, there is a form of active transport that moves both sodium and glucose through the same channel, and that's known as the sodium glucose transporter, which we will focus on in a later video. Okay, so we talked about channel proteins. Channel proteins are like tunnels that allow molecules or ions to move across the plasma membrane. There's another protein known as a carrier protein, which transports entities across the membrane by means of a conformational change. And a conformational change is merely a change in shape of the protein, not to be confused with denaturing of a protein. Denaturing of a protein is where the whole protein breaks down and it's totally non-functional. A conformational change is often what occurs to open up channels, close channels, and in this case, it is how carrier proteins proceed with their work. So this is what happens here. So we have a carrier protein that spans the plasma membrane. And keep in mind that we have solutes out there. So here's this orange thing is representing a solute. Could be sodium, could be glucose, could be any number of things. This is a very common way, actually, how amino acids or tripeptides, which is a chain of 3 amino acids, get across plasma membrane, specifically in the intestinal lining. But when that entity binds to the carrier protein, and there will be a binding site for this, it's going to induce a conformational change where this carrier protein now flips and drops that entity, let's say it's the tripeptide, into the interior of the cell. So initially it was facing towards the exterior of the cell, towards the extracellular fluid. Now it's facing towards the intracellular fluid, and it drops off what it is transporting. And lastly, let's talk about active transport briefly. We will talk about this in much more detail in a subsequent video. But active transport is moving substances against their gradients. So in this case, moving sodium out of the cell from an area of low concentration inside the cell to an area of high concentration outside of the cell, that's moving against its gradient. It's moving from low to high. And I try to represent high and low concentration by having the large sodium in brackets outside, suggesting a high concentration of sodium, and a low concentration of sodium inside, indicated by the smaller Na plus with the brackets. Sodium is moved from the inside of the cell to the exterior often, daily, hourly, by the minute, but that requires energy. It is active transport because it's moving against its gradient and it requires energy. There are two forms of energy utilized for active transport. One is chemical energy, the other is kinetic energy. Chemical energy generally is going to be using ATP. It could be using GTP, but by and large in this class we're referring to ATP as a molecule that confers energy, the chemical energy to carry out active transport. Kinetic energy can also be utilized. It doesn't matter what type of energy it is, but it's either going to be chemical or kinetic. And kinetic energy is the energy of motion. Entities moving down their gradients create kinetic energy, and they're moving based on kinetic energy. For example, sodium moving down its gradient into the cell is creating kinetic energy. And that can be utilized, that kinetic energy can be utilized to bring things into the cell along with it that are moving against their gradients. And we'll talk about this in detail, but the example I've already given is the sodium glucose transporter, where in some cases there's a high concentration of glucose in cells. So to move glucose into the cell, that's moving against its gradient. If it's moving against its gradient, it requires energy. It uses the kinetic energy of sodium moving down its gradient, and that is known as active transport. Specifically, that is a form of secondary active transport, which we will focus on in a later video. Okay, that's it for the primer into transport. We will detail this in more videos to come.